Hello, everybody. Today we'll be looking at the bacterial endotoxin testing. As we can see, I am your presenter, David Ballard. There's also a European presenter that presented on this earlier, so we have Magdalena that worked on it with us. But today, our agenda, we will be looking at the introduction to BET testing and the related standard and international and associated references, the related definitions to the testing, and the basics behind the endotoxin testing. Additionally, we're going to be taking a look at the interpretation of results and what to do in cases of out of specification or out of limits investigations. We'll also be looking at some case studies. A review will showcase practical solutions and some worked examples. And finally, we'll summarize and provide an outlook, followed by a Q&A session. So here on this slide, we have our related standards and references. The first one there, the ISO 11737-3. It was newly published in 2023 and shares many alignments with the ANSI/AMI Standard 72 and details requirements for the bacterial endotoxin testing. The three USP, United States Pharmacopoeia standards, monographs 85, 161, and 1085, all provide guidance on testing approaches for pyrogens, both bacterial in source and other pyrogens associated with products. The ANSI/AMI Standard 72, it includes test methods, requirements for the routine monitoring, and alternatives to batch testing. Then we have several FDA Food and Drug Administration documents, and these range from guidance to industry, review submissions for 510Ks when your device is labeled as sterile. Additionally, we have the 21 CFR Part 820 for quality system requirements. And then lastly, we have our ICH, the International Committee on Harmonization, their guidelines from Q4B, Annex 14. It gives scientific guidelines for bacterial endotoxin testing. Some important definitions for BET include the term itself. BET is the preferred nomenclature. It's bacterial endotoxin testing. Other terms that can be used to describe this or have been used to describe this include LAL or pyrogen testing. The next one we have on our list here is endotoxin. So the part of the endotoxin there is bacterial endotoxin. This is also known as the LPS or the lipopolysaccharide of the gram-negative bacteria. So the next one we have here on our list is LPS. To follow up on that, that's the lipopolysaccharide, the component within the cell wall of the gram-negative that's providing the reactive material and would be causing the fever reaction in people. The next one we have here is pyrogen, and a pyrogen is a substance, i.e. LPS, that is causing the fever reaction. And for bacterial endotoxin, LPS is our culprit. The next component we have here is a concept of maximum valid dilution. This is the bacterial endotoxin detection limit and is based on either a sample dilution or the amount of extract volume that's used when testing products. LAL is our next term here. This is an acronym for limitless amoebocyte lysate. This is used to estimate the endotoxin levels in BET, and in generic terms, it's referred to as just lysate. The next term that we have here is a new acronym that we're using for the water for extractions. And this is an acronym WATER for bacterial endotoxin tests. Historically, it's been referred to as LAL reagent water. And this is a water that we use that will not be reactive with the test itself. Our next concept here is method suitability. It's also referred to as inhibition or enhancement testing, and it's used to determine if there's any interfering factors that will cause inaccuracies of the BET test. The last definition that I want to go through here is lambda, and you can see the symbol there for lambda. Lambda indicates the sensitivity of the LAL reagent. It's in terms of EUs or endotoxin units per ml. It also refers to the lowest endotoxin concentration when we create a standard curve that would support a chromogenic or a turbidimetric method. Okay, moving on from definitions, we're going to take a quick look at pyrogens and bacterial endotoxins to kind of understand what they look like and where they're coming from. So as we had mentioned, a pyrogen is a substance that can cause fever reaction. Predominantly when we look at bacterial endotoxin setting, these are primarily our endotoxin units from either bacteria, could be E. coli or Pseudomonas type bacteria. Here we have a depiction of a gram-negative bacteria, and those little chains hanging off of the bacteria are a representation of the LPS unit, the lipopolysaccharide. It has three main components associated with it: the lipid A, an outer and inner core, and then an O antigen. And while these can vary from bacteria to bacteria, the main structure remains the same. It is what is being detected when we talk about the lysate binding to the LPS here and creating our reaction. So these gram negatives happen to be quite ubiquitous in the environment. Any manufacturing process that includes microorganisms, water is a big factor within the worlds of these bacteria, and it can present itself on forms of biofilms, any tissues of animal origin. So is another predominant source of these gram-negative bacteria, which are leading to endotoxins. So the next thing that we want to cover is the horseshoe crab's role in bacterial endotoxin. So before the horseshoe crab, we had other test systems, animal test systems they would use for bacterial endotoxin. But in this one, we're using an extract from the horseshoe crab. It's the blood itself, an extract from the blood itself that is getting our LAL reagent. In this case, when we're able to use the horseshoe crab's blood, we get a highly sensitive test method that's able to form a clot. And this clot that's being formed is in a similar manner as to how the horseshoe crab's natural immune defense system works to create a clot and, keep it healthy. So the next thing that we want to look at is some principles of BET. And the first concept here that we have is a selection of products. So the selection of products, first would be products of the intended use and how the product will be used in a clinical setting. Products can be with a label claim that they're non-pyrogenic, or maybe a fluid path is non-pyrogenic. Individual sample products can be tested in one of two ways. They can either be done individually or as a pooled sample. As a pool of samples, we have a small disadvantage of hiding variability. You wouldn't be able to see that quite as well. But there is an ease of use from the pooling effect. You can take a pool of, say, 10 samples and test them as one test, which can be cost effective and a little bit easier to manage. So the last point here that we want to put in is that water is commonly tested as a routine sample from a manufacturing process, as the water can host the bacteria of concern and is used for some sort of monitoring control. So for BET, there's a couple different sampling requirements. There's two general approaches that are followed. The first being batch testing. In this case, each lot of product is tested, or those products that would be uniform in character. The second approach here is an alternative to batch testing. In this case, alternatives to batch testing would be used after demonstrating control of materials or the production process. In both cases, we'd be looking for a sampling plan that would be supported by an appropriate risk document to define when sampling should occur and how it should occur. In addition to that, samples are typically analyzed post-sterilization. However, in some cases, pre-sterilization may be acceptable for products that don't support microbial growth. Moving on in principles of BET, we can review an example of a testing scheme. So first is the product handling. Select portions of the product are prepared for extraction to simulate the use. In this case, we can see that happening here. The next step is an extraction of the product. We would use sufficient portions of water for testing, but not more than what would over dilute the endotoxin levels and produce artificially low results. The third step here is the addition of lysate, which dilutes the product a bit further with the test extract. And then we'll follow up that with one of the three methods to approach testing. The first one being a gel clot method. The second being kinetic methods. These would either be turbidimetric or chromogenic. The third approach could be an endpoint method, and this would be a chromogenic photometric method. So the next principle that we want to talk about here is product handling and the endotoxin extraction, and dive into that a bit deeper. Special handling of the product allows for the endotoxin extraction. The first thing that we need to consider is depyrogenation of the toolware and the glass that we'll be using for the extraction. In addition to that, any materials that are in contact with the test system would need to be non-pyrogenic, like your pipette tips. The second thing to consider here is the product-specific endotoxin limit. This is important for the lab in order to determine the maximum valid dilution calculation, which relates directly to the sensitivity of the test and the detection limit. Extraction of solid products can include agitation steps, can include stable conditions at 37 to 40 degrees C. Specific handling times will support how the product is used or possibly what parts of the body the product is used in. And then it's important to note that if we have a liquid product, maybe water from a sample on the production floor, those wouldn't require the elaborate extraction techniques and they could be tested as is. The next thing we'll look at are the BET methods. 3 methods are generally accepted for BET analysis. The first one being, hang tight. Okay. The first one being our gel clot method. This one relies on the formation of a clot and is generally considered a qualitative method. The second one being an endpoint method. This is chromogenic photometric. It's typically noted as a quantitative method and is based on color change. The third known methods are the kinetic methods. These include both a chromogenic and a turbidimetric method, and they both rely on optical density to quantitate and give our measurements for BET in products. After we have the analysis, we're going to take a look at the interpretation of our results. So here's some criteria that is outlined in the ISO standard. But it's important to note that our acceptance criteria can be understood depending on the product-specific BET limits. And criteria for standards is important, as well as negative control and the positive product control. And we can see these standards here include both our gel clot and kinetic and endpoint methods. However, there is some differences here, but they both have criteria set for the quality control of the test method. So we want to take a closer look at out of specification limits and what's needed there when products exceed the limits required. The testing laboratories responsibles include the acceptance criteria, any retests, possible investigation into what happened with the test or the testing requirements. And there's additional guidance that can be provided for out of specification limits outlined in the standards. As we can see here, there's a little bit of infographic that we provided here. Retesting can be done. We can look at the next steps here on out of specification limits. So we have we confirm that we have out of specification. Manufacturer investigates initiates an investigation, and eventually this leads to a root cause finding. And we will eventually get down to a point where we have product that has succeeded and can now pass. This whole root cause analysis is an important step in any out of specification limits. An important part of any BET program, though, is also the method selection. And when determining the appropriate method, the following factors are important to consider. First being the nature of the product sample, the laboratory's capability, the laboratory's experience, any sample throughput requirements, and data handling requirements. As we will see in the following case studies, method selection can be driven by the nature of the product mostly, and how the product itself interacts with the test. This is the first case study that we're going to take a look at. This product is an example of an antimicrobial wound dressing product. We can see there that the bandage is coated with some sort of calcium carbonate coating. Some of the issues that were encountered with this product were high levels of interfering glucans. This created an elevation in the endotoxin levels as it interfered with the cascade of the reagents. Additionally, we had pH levels that exceeded the test range of 6 to 8. So that was causing interference, and the calcium carbonate and related fibers and particles from that were impacting the color and turbidity that also interfered with the testing. So as a lab, we implemented a few things. One of the factors that we can do is a buffer that interferes or blocks the glucans, the interfering factor. Dilute the sample. Several dilutions can be performed. However, it is important to not exceed our maximum valid dilution, and that way we can ensure that the reporting of results is accurate. Another possibility would be to look at adjusting the endotoxin limit value to EUs per gram. That way you have the right weight basis for reporting results, and to choose an appropriate BET method that would support the testing. So when it came down to it, the gel clot method proved to be the successful method. And any future improvements could include the implementation of the recombinant method, as this may bypass the interference that was seen with the glucans. Our next case study is on a black ink or some sort of color marker device. And you can see our example there. So a couple issues here. One was the dark color of the ink didn't allow for a quick dilution to prevent seeing the dark color and the turbidity that caused. The second issue that this causes interference with our turbidimetric methods and chromogenic analysis, as the color would inhibit chromogenic or turbidity from being observed. So some of the workarounds for this type of sample is to understand the customer limits to determine the best dilution, which would be the maximum valid dilution for the product. Again, dilute itself, so we can always look at dilution. And then there might be the possibility of adjusting the endotoxin units to EUs per ml. And upon success, the gel clot method was proven to be successful along with a dilution. As the gel clot method, it provides the similar immune response that we see for horseshoe crabs. However, with the gel clot method, we do get that pass-fail result, more qualitative data than quantitative. Our final case study here is to detail a little bit on manufacturing process water. So process water is taken from a manufacturing process that supports the production, as bacterial endotoxins like to be in water. The culprit organisms will grow in biofilms and contaminate water sources. So some of the things that we do as a lab is we look at adjusting the endotoxin limit to consider the product risk. We could set units to EUs per ml and also consider the actual product limits that the water is in contact with, or family of products. We would definitely use the customer limits to determine a maximum valid dilution. Although the customer limits could be related to the product limits, there could be some other considerations there for QC control. And then the process water is a direct path to testing, as there is no extraction required. And it's a simple dilute, analyze, and then determine the validity of your test. The most successful methods with these are kinetic methods. They've been proven successful, and they provide numerical data that can also be used for trending considerations. Another consideration for process water is the bioburden of the water source. So if you're doing bacterial counts on the water, you can link this up to your endotoxin results and possibly look at further enhancement of that linkage by doing some identification work to understand exactly where those bioburdens are coming from. Other considerations include representative sampling techniques. In that way, you collect a water sample that mimics what the product would see. That way you get your best result from doing this type of monitoring. So lastly here, we have history of the BET with a legacy of an animal-based testing. This has kept patient safety as top priority, but the future looks for an endotoxin test without the animal portion, which brings us to this slide here on recombinant reagents. These recombinant reagents mirror the animal-derived agents and allows the same cascade of reactions with an added benefit though. So we have a small kind of cascade reaction you can take a look at here. On the left there is our traditional LAL reagent cascade, and the middle and right one are the new recombinant factors. The advantage of these is the glucan that can be present in some products and would interfere with the cascade reaction from the traditional LAL sense are no longer causing a reaction and interfering with the traditional LAL path. So these recombinant factors have that one added advantage here, in addition to no longer being of animal source. So these new diversified capabilities are offered up at our STERIS Brooklyn Park lab, if you'd like to learn more. This system is still under investigation, is becoming more accepted though within the standards committees, and will continue to advance and become more commonplace as testing methods continue.